Good afternoon, sir.
Good afternoon.
कसे आहात सर?
किती participant झाले?
Good afternoon, sir. Sir, can we wait another 5 minutes?
Yeah, no problem. No problem.
Good afternoon, one and all.
You're audible. Yes, you're audible. You're audible.
Yeah, thank you. Welcome back, distinguished guests, delegates, ladies and gentlemen. With the kind permission of chairman of today's function, Dr. S.B. Bari sir, I want to proceed with this third session for the national seminar. May I, sir?
Yes, yes, you can proceed.
Thank you, sir. Now we will proceed with the third plenary session of today's national seminar. For this, we have eminent personality, Dr. Ganesh U. Chaturbhuj, sir, to guide us for this session. For this, I request, sir, you to accept our virtual felicitation.
Sir, please accept our felicitation.
Thank you, sir. It is my privilege to introduce Dr. Ganesh Chaturbhuj, Professor, Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai. He is one of the men of distinct vision and full of illuminating ideas and idol of knowledge, experience, and inspiration to all of us. Sir has about 21 years of experience in academics and industry at various capabilities. Sir did his BPharm in 1998 and MPharm in 2000. Dr. Ganesh Chaturbhuj, sir, obtained PhD in pharmaceutical chemistry from University of Mumbai in 2012 under the able guidance of Dr. K.G. Akamanchi, sir, while working as an assistant professor at Institute of Chemical Technology, Mumbai. He joined the group of Dr. Ganesh Thakur at Northeastern University, United States of America, as a postdoctoral fellow in medicinal chemistry under University Grant Commission Obama Singh postdoctoral fellowship during 2013-14. He has served RPG Life Sciences as a research associate in chemical R&D. His research essentially focuses on modeling synthesis and bioactivity of novel molecules for cannabinoids, COX, SGLT2, and TNF-alpha receptors. He is an enthusiast in developing various eco-friendly and economic catalysts and reagents for efficient chemical and heterocyclic transformations. One of such very versatile boron-based catalyst is sulfated polyborate. Sir has published more than 50 research papers in journals of repute, out of which 15 papers are published in tetrahedron letters, and he has 3 patents to his credit. Sir has guided 25 masters and 7 PhD candidates successfully. Dr. Ganesh sir has completed 5 major projects of more than 45 lakhs and many awards to his credit. With the long list of achievements and glory, I would like to invite Dr. Ganesh Chaturbhuj sir to put a light on the topic, importance of organic chemistry practices in drug design and discovery. Please, sir.
Thank you very much. I could achieve you have mentioned. Only because of my teachers.
Sir, your voice is very low actually. Sorry, sir. May I disturb you? Sorry to disturb you, sir, but your voice is not very much clear.
Can you hear me, sir?
सर आवाज येतोय का? सर तुमचं माईक म्यूट करून. Yes, please, sir, proceed.
Hello. Can you hear me now? Hello. Hello. Hello.
No audio, sir, actually.
Hello. Can you hear? Hello.
No, sir, no audio.
Can you hear? Hello?
Yes, sir.
Audible? Yeah.
Yes, sir.
You can clearly see now. You can see my. I will share my things. Okay. I want to
Please, sir.
share.
You can share your screen, sir, please.
Can I share all this complete screen?
Yes, sir. Yes, sir.
Fine. Yeah. Is it visible now?
Yes, sir. It is visible.
Okay, welcome all. I hope the candidate who are around are from pharmacy and as well as chemistry background. Yes, sir. Correct?
Yes, sir.
Yes, sir.
Yeah. So basically what I'm going to cover today, I would say it is more informal in that way, because the thing we make something formal become difficult to assimilate. So I'm going to discuss today importance of organic chemistry practices in drug design and discovery. So you must be already been taught or discussed with other speakers about what is drug discovery, what is drug design, but something which is I can say it is not incomplete, but it is not in real sense coming into practice. Being a pharmacy student, we know that the organic chemistry content is very limited in our syllabus, and that ends at almost second year, and at the most third year we do some small drug synthesis in the laboratory. What we need to understand, because we are going to produce something which is not reported in the literature, and these molecules, unless they are structurally different, do not have any CAS number, and do not have any kind of earlier reports on that, then it is patentable, it is novel, and then it has acceptance in the journal and the patents and all. So one need to, along with the knowledge of biology, uh, knowledge of various your biological technique systems, receptors, different biological processes, and biochemistry, you must also know how to make your own molecules. If you are unable to make your own molecules, you will be dependent on the organic chemist to make molecules for you. And then you have to disclose the structure of compound to him, and then there will be a infringement of the patent, and sometimes that data will be getting leaked before you put patent or a research paper. So I'm going to brief in a small way what is medicinal chemistry. Basically, what we try to do in medicinal chemistry, the medicinal chemist is actually applied branch, a combination of biological science, organic chemistry, biology, and computer science combinedly forms a medicinal chemistry. It medicinal chemistry is like its origin is trackable up to genetic mapping and protein crystallography. So unless some basic research done by biologists, medicinal chemistry doesn't start nowadays. Earlier, before invention of software, before invention of X-ray crystallography, before invention of any systematic approach, the entire medicinal chemistry was based on serendipity. A serendipity means a chance, which is called as a luck. They were developing the drugs based on the intuition. Intuition means when I sleep in the night, I find some structure. Same like Archimedes went out of the bathroom without clothes, saying "Eureka, Eureka." It's like that. So if I'm sitting alone and just doing some kind of meditation, I get a dream that this molecule could be more potent. So that was the way to develop the drugs, and rather it was now considered to be a disadvantage that you have to make so many molecules and then you have to test all of them. And then unless you test all of them, you cannot get which compound is pure, which compound is more active, which more effective, non-toxic. That can be only done after this. So what we generally do, the medicinal chemistry chemists have to have a comprehensive knowledge of biological targets. It can be organ, it can be receptor, pharmacological actions mediated by the receptor subtypes, what is their similarity and differences, biological substrates for the receptor. For example, there is an endogenous ligand that binds to the receptor, and which should be known by the medicinal chemist. Based on that, some discoveries can be done. So you must be knowing there are many anti-metabolites, competitive inhibitors are developed based on the structure of the natural ligand of the receptor. For example, sulfonamides are displacing para-amino benzoic acid from the site of DHFR enzyme. So this is one example of biological substrate. And list of all the molecules which are active against that receptor, if they are in the literature. You also should have knowledge of computers, software, hardware, which kind of software will be suitable, what kind of hardware shall I buy, what should be the capacity in terms of computational power. All those things should be known to the medicinal chemistry. But in addition to this, I'm just looking at chemistry. I would say there is nothing on this earth that is made by the man is without chemistry. So that is my belief that everything that is man-made has some chemistry. Let it be anything, anything. No, you take examples here. Paper which we use, cellulose, has like you have to cellulose. You get a brown from the plant. You convert into white paper, red paper, blue paper. That's the chemistry. Cosmetics, nail paint remover, nitrocellulose, and butyl acetate. Candles are wax. Okay. Fabrics. All the fabrics are nowadays are synthetic because that much cotton is not available to have everyone cotton clothes. One time the synthetic fabrics were costlier than the cotton. Now the cotton fabrics are costlier than the synthetic. It's all market depends upon the availability. Soap. Before invention of this synthetic soap, it was all natural soaps like shikakai, Rita, and all those we were using in earlier days. But when the modern era, you are using soap. Not only this soap, but soap for everything. Soap for face is different. Soap for cleaning is different. Soap for clothes are different. Soap for bathing is different. Soap for hairs is different. Shampoos. Okay. Now the gasoline. Gasoline means the hydrocarbon that is we use for either in the petrol tank or what we use it in the household as a gas. Now, if you look at the real life application, naphthalene, it's a chemistry which is naphthalene balls which you put for deodorization in the cupboards and the toilets. Eugenol, which is in the clove, which is everyday, you can say, our condiments, masala in the kitchen contains clove, and for toothache we use it. Cinnamon, again a part of whatever spices we use, and still it has a chemistry. So without chemistry, you cannot live. Now, these are a few more examples where organic chemistry is useful. A cosmetic, agrochemistry, you name the thing, everywhere there's a chemistry. Medicines, cosmetics, wine, anything that is chocolates, canned food, preservatives, you cannot escape. All consumer and consumer durable goods, including paints, varnishes, automobile parts, electronics, everything is chemistry. And most of the time you find it's very less inorganic, but it is more of organic chemistry. So you cannot escape out of this. So I just wanted to give a glimpse where the chemistry is penetrating up to our household and daily use products. Now, organic chemistry, which is called as man-made chemistry, this is used for man-made preparations. Now, all polymers, let it be used for electrical purpose, let it be used for thermocol, let it be used for uh, electronic gadgets, everything that is plastic is man-made. So basically what we do in organic chemistry, it's a basic science, deals with various chemical transformations of hydrocarbons. Why am I calling it as hydrocarbon? Because that's the source that is coming from the earth crust. We take a crude naphtha and isolate the compounds and we convert them into different forms. Using reagents and catalysts, reagent catalyst can be inorganic, organic, or organometallic. Different organic mechanism of reactions we understand by organic chemistry. We understand isolation, purification, we understand structural interpretation. So there is something which is instrumental analysis also touching organic chemistry, because once you make the compound, you need to confirm the presence of that. So you also have one part of analytical instrumentation. Not I'm talking about details of instrumentation, but interpretation is part of organic chemistry. Hence, everything that we see that the man made is called as synthetic. And whatever comes from the nature, we call it as natural origin. So, in turn, we derived from natural origin hydrocarbons and organic chemistry efforts we do on that. So, list is very long, ranging from medicine, clothing, consumer durable, consumer goods, everywhere the organic chemistry is already has a presence. Now, what chemists see the dream, what medicinal chemists see the dream means actual dream or intuition, that the molecule which I am working on, whether they are natural, semi-synthetic, synthetic, whether it is biological origin, biotechnological origin, it should see the light of the day. That means it should be coming in the use and reducing the pains and sufferings of human being. So medicinal chemists dream for that there should be increased efficiency or selectivity compared to the one which is reported, reduce toxicity, increase ADME properties, that is solubility and bioavailability, reduce metabolism, increasing the half-life, and obviously sometimes we look for targeted activity. Means non-selectivity can be reduced, dose can be reduced, drug can directly go to the site of action where it is supposed to go. So that is what the medicinal chemist is looking at. But how he can achieve this? This can be achieved only by mastering organic chemistry knowledge. Otherwise, the whatever software technique we use will remain in a theory. It will never come into the practice unless the medicinal chemist convert their knowledge which he gained from the design part into the actual material produced in the lab, which is pure, well characterized, and then applied to the different receptor systems for biological testing, and then he gets some results, and that results can be finally interpreted in terms of this. So all molecules, either peptidic means polypeptides, non-peptidic, natural, semi-synthetic, synthetic, are the derivatives of hydrocarbon. You name the compound, let it be uh, carbohydrate, let it be proteins, let it be any synthetic product which is there. They are derivative of hydrocarbon with or without heteroatoms. So there can be that, maybe heteroatom, may not be the heteroatom. So there are some compounds which are without heteroatom, only carbon and hydrogen. Most of the steroids contain only oxygen, rarely nitrogen in them. So they are more of hydrocarbon nature. And there are compounds which are heterocyclic in nature, more aromatic in nature. So organic chemistry knowledge is needed to design the molecule till marketing, post-marketing surveillance. Everywhere organic chemistry is needed. Now to design the molecule, you may get confused. Why we need organic chemistry knowledge for this? We'll be discussing in due course of time. To realize the dream of medicinal chemist from the one C by intuition. Intuition means by my previous experiences, I get some inner voice that I must do this change so that I can, this will be active. Predicted means you do the computational study and then predict. Postulated means without any basis, you say that yes, this will be activated, this will be available. So you can get all this extrapolated, calculated, everything you can do by software to clinical use. That's what the dream is. So everything what you predict, you intuition you do, postulate you do the extrapolation, that should finally turn into, should go to the clinic. That should be near the bedside of the patient. It should be used clinically. And that largely depend upon organic chemistry knowledge. So we'll discuss how it is, how organic chemistry is playing role. Now, serendipities and systematic and iterative processes. What is this? Serendipity means I am writing in Marathi, it is nasib, or it is chance. It is by chance. Okay. Before systematic approach, all the discoveries which happened were either from natural origin or by serendipity discoveries. Okay. And while predicting the activity using systematic or in silico methods, serendipity method, medicinal chemist has to select a series of molecules. Now you select the series of molecule and then put them either to the serendipity or you put them for systematic approach. So ultimate aim of the medicinal chemist to investigate the effect of structural changes on potency, selectivity, efficacy, toxicity, absorption, distribution, metabolism and excretion, solubility and metabolic stability. So these are the aim of the medicinal chemist. The drug which is going to go for biological testing in animals should have proper potency, selectivity for one particular receptor, higher efficacy, low toxicity, better ADME properties, good solubility, because most of the time the drug is effective, but it is not soluble. That's why it is not showing activity. So based on the experiences with the series of the molecule, decision need to be taken for further iterations. Iterations means you repeat the same thing again and again. So what do we do? You do make a molecule of say about 25 molecules you make and subject to biological test. So you get certain responses that some compounds are soluble, some are not soluble, some are more active, some are less active, and you come back and redesign your structure, resynthesize, then retest, and this process continuously happening. That is called as iterations or iterative design. Iterative design means it is always in feedback with the biological activity. Once the biological activity is improving, you go in that direction and changes your structure in that fashion. So till the desired target of potency, selectivity, efficacy, toxicity, ADME, solubility, metabolic stability is achieved. So it can be all the aims together, or compound may be very selective but not soluble. So your target is only solubility. Your compound is soluble, good ADME, good efficacy, but metabolic is not stable. So your target is metabolic stability. So these all the target may be together or they may be independent. Okay. So medicinal chemistry is an iterative process, balancing between the desired attribute. Desired attribute means whatever we discussed earlier, and chemical or structural features of the molecule. So you keep on juggling. Juggling means how you take a three balls and keep on bouncing, and you have to keep all of them one time at hand, and you keep on juggling with various chemical structural features to have desired biological activity while not compromising on toxicity and ADME properties. So there, and the software tell that it's ADME property, toxicity property, toxicophores. Those are all available in the softwares. But ultimately, time is the money. So while doing this, one have to keep in mind that the timelines and validate the direction of work, where you are going. Okay, whether you are going in the right direction or you may have to come back. So sometimes we find the drug is very effective, but unfortunately, as per the ADME are concerned, it falls in BCS class 3 and 4. So you are aware that what is BCS class 3 and 4? That BCS class 3 and 4, they are highly soluble, low permeable, or low soluble, low permeable. So that is why if this happens, the efficacy wins over ADME, and the pharmaceutics scientist gets a new target molecule to handle. So you must have seen most of the pharmaceutics scientists around us keep on working on solubility enhancement, targeted delivery, nano delivery. Why they are doing it? Because your compound is so active, so potent, it causes toxicity. You need a selective delivery. Your compound is not soluble; it coming in BCS class. But the actual target for any of the medicinal chemist is to have all the molecules in BCS class 1, but it never happens. It never happened in future. You get efficacy, but you don't get BCS classification, or you get BCS classification, but the drugs are not that effective. So you have to have a dose level as low as possible, and then you should have a good permeability, good solubility. So that can be achieving these two are not very easy. So while designing the drug, ADME properties, biopharmaceutical classification is also taken into consideration, but that need to be practically proven. So what organic chemistry is all about? Organic chemistry is about name reactions. Organic chemistry is about reaction conditions. Organic chemistry is about reagents and catalysts. Organic chemistry is about regio and stereo selectivity and the yield. And it is about the solubility and stability of the molecules is equally important. So considering organic chemistry, it revolves around this. And there are many more aspects of this, but this is for this today's discussion. This is what I think is about the discussion. Now, what is the role of organic chemistry towards medicinal chemistry? What exactly we try to do? I'll be discussing one point with very limited examples. Understanding chemical space, that is called as libraries, and the structures. What do you mean by libraries? There are many databases available which can give you the type of structures which has activity. like a zinc database, and there are many more databases available, which can be utilized for virtual screening or in silico screening. So we need to understand what is that chemical space. Chemical space is nothing but what kind of structural features do you look for. Okay. Then important to understand biomolecules, that is biospace, and interaction of molecule, that interaction of chemical space with biospace. So that we can find out by different thing. But what is the connection of this? You may say that this is like same like a drug discovery or using say softwares for the purpose of deciding which molecule can be prepared. No, to understand which molecule to be chosen for the synthesis, which molecule can be active, can have some kind of interaction, you should understand the chemical structures. And to understand the chemical structures, to understand the biomolecules, ultimately, what are the biomolecules? Biomolecules are nothing but they are polypeptide, they are receptors, and they are connected with some biochemical route which it is following. So you need to understand chemistry of the structures and chemistry of biomolecules. And then only if you have organic chemistry sound, surely you can understand these things properly. The most important part which we generally come across in medicinal chemistry books called as SAR, structure activity relationship. Now, what we try to do in structure activity relationship, we try to change the structures, vary the structure, and what we try to keep the common thing is homologous series. So central core of the molecule remains same, while the entire substrates around, or you can say substituent around, that will change. R1, R2, R3, R4. You can have variants there, and you keep on changing those structure. Now, my question arises: you may design anything on the computer. You may design everything on the software. Do you Are we sure that we make that in the lab? So to understand whether whatever I am predicting, whether it is synthesizable in the lab or not, if that knowledge you have, then your molecule which you design will be synthesizable, and you will design something which can be done. Otherwise, you can attach any molecule to any molecule for the sake of activity. You may not be able to produce that in the lab. So understanding organic chemistry for SAR is equally important. Addressing metabolic stability by chemical modifications is also important part, because metabolism is a chemical reaction. Only thing that is organic transformation only it is done by the enzymes at room temperature in the liver, in other places in the body, and they are modifying my structure so that its activity will go away, or it will turn very quickly into soluble form and will be thrown away by excretion. So to addressing that metabolic stability, one should understand what chemical change or whatever chemical reaction that enzyme is doing, so that how I can modify. Now, the classical example of this is a replacement of hydrogen by deuterium. What is the change? It is hydrogen only. One neutron or one positron is higher. So the atomic weight is 2 instead of 1. Now, in the nature, the presence of deuterium is very less, and biological molecule do not contain any deuterium. So the enzyme which is metabolizing at that particular point, if that H is replaced by D, enzyme gets like surprise. Molecular size increases, atomic size increases, and it cannot fit into that, and it has no information, or there is no coding that can help it to modify itself to remove the deuterium. It can remove H, which is naturally present, but it cannot remove D. So that's why you should have the biochemical pathway and how we can understand that biochemical pathway, and accordingly you can make changes in the functional group which are liable for metabolism. Then observing metabolism, effect of metabolites on physiological, pharmacological actions need discovery of the new molecules. For example, hydroxychloroquine, which is currently used for COVID, is actually a metabolite of chloroquine, and hydroxyendomethacin is also active metabolite. Rather, endomethacin is not active, but hydroxyendomethacin is more active. So one can study. You know that it is actually a synthetic, a bunch of enzyme in liver are going to first attack on the molecule the moment come in. So any molecule, any drug that is consumed, any chemical that is consumed will be first attacked by the liver enzymes, and it will start making its derivatives. We suppose that the molecule which I am putting into the body is more reactive, or because of that I am getting activity. No, there are something, you get some side effect, you get some ADR because of the metabolite that is generated by. That is why the fixed dose combination were sometime in a still there in the question, that if I fix 3 or 4 drugs together and I put them in the liver and the liver start breaking down them, how many molecules it can make? And those all molecules, whether they have some interaction with each other. So one need to study that, and then from that you can come to know, yes, my hydroxyendomethacin is more active than endomethacin, and because of the metabolism my activity is increasing. So to understand that what chemical change has happened, organic chemistry knowledge is important. Now the most common approach was there, like designing products. I can leave this PPT with you, where you can go through this website, this particular paper in Nature Review, and you can read that how products are prepared and how products are important. So I will discuss that in a coming slide. We make a products like this. What do we do? You take a drug, you attach something, the group that enters into the body, acted upon by the enzyme, remove this part, drug is released, and then act. So you can increase metabolic stability, you can increase absorption, you can increase so many different characters by putting drug into product form. So what are those groups are? Carbamates can be made, amides can be made, you can make manic bases, you can make ethers, you can make phosphinates, and number of different things are. Ester is very common kind of, you can make different types of prodrugs in ester form. So there are examples available. Tagging drug to the biomolecule. So this is very common practice in pharmaceutics. That suppose I got an antibody against a cancer cell. So what I would do, I would tag my drug to it. What do you mean by tagging? Will it just go and sit on it? Whether it will go and just adsorb on it? No. There is a chemical transformation. There is some chemical bonding or covalent bonding that need to be done with the conjugate, which called a conjugate, which can be with the biomolecules which are going to go and only sit on the cancer cells. You have to chemically bind your molecule to that. So there you require what is the structural feature of biomolecule, where I can tag, what kind of linker I can use, what kind of reaction I can use to tag this. So that is very important. Targeting the molecules. The most important, you already aware that about 40% of the molecules in pharmacopoeia or in use are chiral in nature. And what is the importance of chirality? Everybody is aware that use of racemate, use of mixed isomer is not allowed these days. Actually, we are a story of thalidomide, which was given in a pregnant ladies. It crossed the placental barrier, which is called as teratogenic effect, and then ultimately enter into the baby, and the baby born with the sealed limbs. So that is the reason since then the chirality and the drug action came into more discussion. And nowadays, not even 0.1% of the other isomer is accepted by the pharmacopoeia. So we have to find it by polarimetry, you have to find it by various techniques like chiral HPLC techniques, by enantiomeric access. So importance of chirality, and then chemist has to design the organic methods to have stereoselective synthetic methods, or you have to design the separations, understanding the chirality. Understanding chemical structure and their susceptibility to light, heat, oxygen, carbon dioxide, acids, bases, moisture, enzymes. Okay. In turn, stability of the molecule. So your molecule which you predict sometime susceptible to any of this, and it loses its activity. So stability of the drug is also important. Unless it is proven as a effective and non-toxic in human beings, it cannot be called as drug. Till that time, let us call it as chemical structure, or let us call it as new chemical entity. Unless the new chemical entity are stable to these things, you cannot store them for long. You cannot apply them to this. So, like understanding chemical stability of azitidine, that is three-membered ring containing nitrogen, azatidine, four-membered ring containing nitrogen, cyclopropyl, all these strained rings are so strained, the bond angle is so tightly packed, it can open at any time. Okay. So understanding stability is important. Understanding stability of natural product means something you get vincristine, vinblastine, you get Raulfia alkaloids. How long they are stable, or can they be converted to stable derivative by a semi-synthetic way? For example, vinblastine from Vinca rosea. Okay, now this is vinblastine. So let me. I think I have some correction here. I wanted to show you the difference, but fortunately, it is something. Okay, right. So there is a formyl group in vincristine, which is reduced to methyl by vinblastine. So this formulation, it would not remain stable. So it is converted into semi-synthetically. Just one look at all the entire structure is same, except in one corner. So if you just want to see that in a slightly expanded way, so you can see that it is formyl and this is methyl. Okay. Now coming to the other one, that is artemisinin. Artemisinin is actually prepared from, it is isolated from a Chinese herb called Artemisia, which is small herb, and now it is done by synthetic way. Now this cyclic lactone, cyclic ester, which is called as lactone, is reduced to alcohol, is called as dihydroartemisinin. Okay, there is no change except this. Now when this artemisinin hydroxy is converted to methyl group, is called as artemether. Similarly, this alcohol group, when attached with succinic acid, monoester, you can say artesunate. And artemiside is replacement of this entire OH by thiomorpholin, and thiomorpholin is oxidized, is called as artemisone. So these are all related structures, but original structure is artemisinin, because this can open up into structure and the stability will decrease. Hence, it is converted into semi-synthetic derivatives by small structural changes, majority part of the molecule, and this is the only drug, artemisinin and derivatives are effective. Also tried for COVID, but earlier it was it's very effective on all four strains of malaria, and cerebral malaria, that is falciparum kind of malaria, which is very deadly, can kill the person. These are the only effective molecule. And the lady, which is Tu Yu, she is a scientist, Chinese scientist, got Nobel Prize for this work. Okay, now this we saw. Now look at the prodrug kind of thing. I was just talking about that. It is bioactivation or breaking down of molecule and releasing the active molecule. Now this is Candesartan, which is antihypertensive. It is angiotensin converting enzyme blocker. Candesartan has carboxylic acid, which is attached with this ester, which is very labile ester, which is cyclohexyl oxycarbonyl oxymethyl. So this particular called as silex-acetyl. It can break very easily by enzymatic cleavage, but till you formulate it, it remains stable, right? So Candesartan is given as Candesartan silex-acetyl, but This is lamivudine, which is the drug, which is actually a cytidine kind of structure. The amino group is protected by formamidine molecule with dibenzyl amine, and then you can have formamidine structure, and then in situ it breaks down and releases the drug. So for stability purpose, it's done. Now dopamine, which is natural, uh, say neurotransmitter, which is given as levodopa. When decarboxylation takes place, levodopa get converted into dopamine. Similarly, sulindac, which is actually this is sulfide, but supplied is sulfoxide, and this sulfoxide as a prodrug action is not there unless this converts back into S. And these are few examples of products. There are many products, phosphoric acid products, ester products. You can refer to that particular uh, nature review. You will come across with that. Okay. Now, what is another thing? What understanding we need is heterocyclic synthesis. Synthesis of thiophene, synthesis of furan, synthesis of pyridine, pyrimidine, synthesis of fused heterocycle like benzofuran, indol, it may be benzothiazole, benzoxazole. There are many heterocyclic synthesis can be done. And heterocycles, why heterocycles? Because most of the body, actually, if you just start calculating, the body is made up of 70% water and 30% a solid part. Out of that, majority is bone. Okay. And the remaining part is made up of proteins. Entire thing is protein. Skin is protein, muscle is protein, hairs are proteins, the enzymes are proteins, the hormones, some of them are protein in nature. So everywhere there is a protein, and we all have L amino acids in our body. And because of that L amino acid, it has a particular twisting or particular folding of that proteins. So we are all made up of nitrogenous material. Phosphorus is the energy source. And when that is going on, you need some heteroatom to interact with the proteins. And the active sites are made up of amino acids, and you require heteroatoms, hydrogen bonding kind of thing. So without heteroatom, there are very few drugs are available. So one need to understand heterocyclic synthesis. One need to understand expeditious, means fast, high yielding method with the catalyst and a green approach. One need to understand during designing the chemical entities about what is feasible, okay, to synthesize, and are there any synthetic method available in the literature to make this molecule. So before you start, when you start designing molecules, just don't go by like a kid, attach anything to anything, and then I may be able to create in future. In silico method, you will get very good activity. But when it comes to the lab, you may not be able to make those molecules. And over the period when I'm working for so long, our idea is that whenever we predict something, either that is not very active in silico, and even if that is very active in silico, it cannot be made in the lab. So one need to use some tools called as a Reaxis and SciFinder. So let me go little to my computer, and I will show you what the Reaxis means. I'm opening this Reaxis thing. Now here, this Reaxis is a search. It's actually developed by Elsevier, which is a publisher, European publisher, and it contains about 179 million substances, 57 million reactions, 86 million documents, and 41 million bioactivities. So if I want to just search, I can put my CAS number, I can put my name, I can put my structure. So let's go, and we can just put here hydroxychloroquine. I'm just typing the name. The moment I put the name, I can get the structure here. This is my hydroxy. This is hydroxy part. And actually, this is metabolic hydroxylation. It cannot be made in the lab. So what happened in the body when this is given, it actually got hydroxylated at this point. So if I go to transfer, I'm transferring it for the query. and this query is being shifted. Now this I got everything about this. I got the substances, I got the document, I got related substances, I got the reactions, and I also got the commercial sources. So I would first go to the substances. I'm going to see what I got here, and I just clearly see that yes, I got so many things. Identification, if I click, I will get name, alternate name, I get reaxis ID, I get gas number, I get molecular weight, I get empirical formula, I get what substance labels are, means what is the alternative names that can be given here. So I can get so many references for label. I also get drug likeliness, I get patent data, I get derivatives, I get physical constant like melting point, absorption, chromatography, crystallography. I can get spectra like NMR, IR, mass, UV. I get bioactivity data, pharmacological activity data. So many references are there. I just go to them and find out patents are there. I can find what for it is patented. So I get everything around that. And I also get after biological activity data. I can just minimize this. Other data is also available, which is uses quantum chemical calculation somebody has done. So I can go through all this data. This is very good database where I can search my compounds. And it is very important to search the compound before you start the synthesis to understand whether my molecules are novel, nobody has reported them, what is my similarity of my molecules with the already reported molecules, whether they have Cas number, but along with the synthesis, you will come to know with that only. So if you want to know how it is prepared, I will just go to the preparations and click, and I will find so many methods. There are 14 methods how it is prepared, and I can see that it is prepared by condensation of this amine with this chloro. So I can at least get some idea. My molecule may be slightly different, but I can get some organic chemistry idea. And I also get a references here. I can go to the patent, I can go to the references and find out. Now this is the journal which I got. The moment I click on journal, I will be transferred directly through the DOI to the journal page directly. So this is my journal page. If you have subscription, you can straight away download this PDF, and it is, you can clearly see how they have prepared this. So I'm directly reaching to the way of synthesizing this, and I know how this fragment is prepared. I know how this fragment is prepared and how they are condensed together. Okay, this arrow stands for a retrosynthesis arrow, a reverse synthesis or reverse engineering. So I must take this, which I'm getting iodinated. I'm going to make this, and then I'm going to condense. So this is the way you are going to come. And then the forward synthesis is given. It is done by the flow chemistry, this reaction, and everything is done by flow chemistry. So you can simply integrate it. Okay, so this is the way the reaxis is used. There is another called a SciFinder. Unfortunately, SciFinder, I have given you the thing here, but our subscription comes from UGC. So currently, government has not renewed that subscription. So this is the issue, that we are not getting access to SciFinder. Okay, Reaxis is available with us. We use. Now there is one more thing which I want you to read, go through this webinar on retrosynthesis. Now let me tell you what is retrosynthesis. Suppose this is my molecule to be handled. Okay, how I can reach to its starting material, a simplest possible starting material which is commercially available, and I keep on converting them one after the other step, and then bring it to the final molecule. So retrosynthesis is the reverse of synthesis. Synthesis means making. Reverse of synthesis, retrosynthesis means breaking. I will break down this molecule into small fractions. Still, a logical starting material can be derived. Unfortunately, not every scientist have that ability to do that. So the Merck group, now I'm just opening this page. Merck group has developed software called as Cynthia, and Cynthia suggests various routes. You can just see on my slide how they are showing it. if I just expand this and I show you, you can see here on the computer, can you see something? What this small red, green, white balls, and what kind of tree you can see? This tree is nothing but the extremities are actually the starting material from which you can start with. And the point is actually the reaction conditions. And then you can achieve the final molecule is at the center. And these are the possible routes that you can apply to bring to your thing. For example, I can give me just 1 minute. I will just want to use some. I always have a habit of, as I teach in the class, I'm not very accustomed to the PowerPoint presentation. I generally prefer using a whiteboard and all, but still it has advantage, obviously. So what I'm trying to show you here, that I want to add one slide here. I wish to prepare simple molecule like benzonitrile. Now, how we can achieve this? Shall I bring it from dehydration of amide? This is one thing, right? I can remove this H and O so that I can get CN. Shall I bring it from halogen here and then maybe sodium cyanide, right? Okay. Shall I bring it that is called as from methyl straight, directly from methyl? That is called as ammoxidation. And this is the commercial industrial way of making this. This is hazardous. This you have to come through again from benzoyl chloride, and this benzoyl chloride is going to come obviously with ammonia. This has to come from benzoic acid. Benzoic acid has to come from oxidation of toluene. So step number 1, step number 2, step number 3, step number 4. While here it is one step, toluene to ammoxidation. It may or may not be possible for all the molecules that you are going to handle. Sometime you may have to come from acid to acid chloride to amide to nitrile, or sometime you may have to come, it's molecule is so complex that you cannot get methyl there, you have to come from halogen. Okay, you can get halogen easily, so just displace it by cyanide. Now the safer options for this are copper cyanide, CuCN twice. You can even use calcium cyanide. Okay, so these are safer sources. Okay, so this is called as retro. If you want to write a forward synthesis, I will put this as a starting material and then I will put this. So this is the way for any molecule that you want to make. There are very comprehensive books are available. Comprehensive heterocyclic synthesis, comprehensive organic synthesis, comprehensive medicinal chemistry books. There are comprehensive volumes are available where they have summarized the data of last 50 or 60 years or one complete century, and you can go through, there are references given and you can use them for your purpose. Now this is very interesting. I advise you to open your mobiles and sir, I can just send this link your mobile, rather shall I send a mobile or email? Hello,
Sir, you can put it in the chat box actually.
that is good.
Yeah.
Okay, so I'm putting in the chat box and you guys can just simply take this. It is in the Play Store, Google Play Store, or those who cannot get this, just type in the Google reaction, Google this one, Google Play reaction flash, and what you find, you'll find this kind of orange structure. It is also supported by Reaxys, which is again elsewhere initiative. And if you download, let me know. Now these are the screenshot of shots of my mobile, and I carry this app all the time because nowadays everybody is busy. Sometimes they're traveling from long distances, and they really get time during travel. So suppose there are 1094 reactions invented so far. Out of that, 37 are new reactions, 83 are updated one, and remaining you can just go through your app, you'll come to know. And they are arranged in alphabetical way, and they are also searchable. You can search where they are. Okay. Now, participant, you can put in the chat box. Could you download it? Any one of you, would you like to answer that in chat box? Or any difficulty?
Sorry, is it only for Android or Windows?
It is only for Android.
Yeah.
But still, so Safari, you can search reaction flash. It must be there because
Yeah.
I'm using Windows. I never searched that, but it must be there.
Okay. Okay.
iOS must be there. So if you already downloaded, you can follow. Click on the tab, the app icon. It will open like this orange, and then it will appear like this. Okay, it will appear like this first. You can click on all reactions. You will find a list alphabetically. Aldol condensation I have selected because aldol is known to all. So I'm selecting aldol condensation. You can also try if you downloaded so far. I think by now, if you have any difficulty, shall I wait for some time? You can answer in the chat box.
Dear participants, is there anyone who requires some time? You can just put in the chat box, or you can just raise your hand and ask the query if it is there.
If you are facing some issue in downloading, let me know. Okay, I shall continue this. Okay. So when you click on the reactions, you will get the list of the reactions. And once you see they are alphabetically ordered, so I'm just going down and finding out aldol, which is marked orange. Okay. And you can just click on that aldol, so you can find what I can see in the third page. So I'm getting what is aldol condensation. So it's telling me that aldol condensation, carbon-carbon bond formation reaction via enolate addition and loss of water to form alpha beta unsaturated carbonyl compounds. So this is what aldol condensation means. First formation of alcohol and then dehydration to get double bond. Okay. Now when I click, there is a, you can see at the bottom, you can see this conical flask. If you click on a conical flask and go to the, you will get a reaction, mechanism, examples, quiz, and you also have looking for in Reaxys. Okay, so click on mechanism, so you can find this mechanism, how this reaction happens. Okay, and you can still go back by tapping on that mechanism again and click on example. You can see this. What the blue color sign are the hyperlinks. The moment you click on this, you can directly reach to the journal page, and that you can see. So directly you reaches to this particular journal. So you can even see where they are applied in practice. So organic chemistry and mathematics need to be taught when, where this is name reaction, this is the compound, and how, where this is applied. Okay, and where these are applicable for drug synthesis. So every name reaction has application in the drug synthesis. So it should be taught. So I am expecting you to download And for this particular reaction, there are 10 references which are there in different types of journals of very high impact factor. And either they made starting material or they made a final product, but they have done something, some catalysts are developed for this reaction. So you can just click on that and just find out how this can be utilized. So this is about reaction flash, which is a handy tool you must carry to understand organic chemistry. Now coming to the next, in short, I am coming to the end, but not really end. In CRISP, what medicinal chemistry shall learn about before beginning the work are name reactions. So you are aware in your syllabus, you have Claisen reaction, Claisen-Simons reaction, Curtis reaction, Aldol condensation, amidation, acylation. All these are name reactions. So whatever I have shown, the reaction flash is actually a store of all the name reactions. So you can read them wherever you are. It is not depend upon the laptop. You can simply carry, and not works on laptop. It works only on Android. Okay, it's not made for laptop. Organic functional group transformations that we'll discuss. As I told you, this is called as functional group transformation. One functional group is converted. This is by oxidation. This is by amidation. This is, you are getting acid chloride, and this is the dehydration. So one need to understand what is the functional group transformation. Functional groups are the group who are taking part in the reaction. Acid, aldehydes, ketones, amines, amides, nitriles, lactones. These are the functional group which can be converted into one another. Reagents used for organic transformations and heterocyclic synthesis. Oxidation, reduction, cyclization, dehydration, chlorination, bromination, iodination, all these transformations and heterocyclic synthesis, you require different kinds of reagents and catalysts. You must know that before you start. Preparation of a compound, stability of compound, safety of the compound, all the reactant and the product, you should understand material safety data sheets, how they are dangerous because they are not food, they are chemicals. Some of them will be silent, solid, non-volatile. Others may be fumigating, irritating, giving irritation to the skin, eye. What kind of precautions shall I use before doing? Understanding their labeling, understanding barcoding, their storage, and you must keep a data. Data keeping is very important in any science. Chemistry is very important for medicinal chemist, how the molecule was made, and you should be able to repeat. Okay. Now MSDS, I have given the link. I also given, now this can show, I can just click on this MSDS. I can see that this is material data safety sheets for, there are different companies who are uploaded. If I click on this, I can simply find they are there. So it need a membership. You can sign up for this. Okay. So you can get this data material sheets. Now these are very important books. I would like to recommend you S.N. Sanyal. There are two books. One is Reaction, Rearrangements and Reagents. Reactions, rearrangement, reagents, by this understanding reaction mechanisms. So there are two different books are available. They are available on Amazon. I have given you the link. Understanding Organic Reaction Mechanism by Adam Jacob. Very important book. I have read this completely. Okay. You must read these books, the recent editions and all. They're available Amazon, very cost effective, not very, see, it is hardly 248 rupees book, which is very important for your organic chemistry knowledge, and you can understand many things out of this. So you'll get interest, very Indian language and very easily palatable. Now I was talking about organic, functional organic group transformation. Okay. One is converted to other. The most important book that I want to show you is called as Comprehensive Organic Transformations. I request the colleges who are working on synthetic chemistry or they are working on medicinal chemistry or any kind of organic chemistry activity. This book should be there. It is 4 volumes are available. They are little costly, but this book suggests what kind of there is a book review. You can see down here. It can be reduction, page number 5. coupling reaction 194, Friedrich-Crafts, ring forming reactions, aromatization, alkenes reactions, elimination reactions, how to prepare the alkenes. Okay. Then Wittig and related reactions, metal promoting coupling reactions, arene, alkyne, diene reactions. There are all reactions are given. There are 4 volumes. You can understand alkynes, halides, amines, ethers, alcohols, phenols. So I can just simply copy this and put it in the chart box so you can access this quickly. And you can see this is about book review about this book. The cost is about $1,200. I would say it is around about ₹1 lakh, 1 lakh rupees 4 volume, but institute can afford and keep in the library. Resolution, alcohol, transposition, substitution, all everything is given about all functional groups. 4 volumes is very comprehensive, and it also suggests you when one group is present how other can be handled. The selectivity in the chemical reactions is also given in this book, along with the references of the journals where it is reported. Very very important book, comprehensive organic transformation by R.C. Laroque. If you can get some e-copy online, you must try. I don't think it may be copyright protected. So this is a very important book for you. The most important after this all you know to set up the reaction, how to put the reflux, how to put the moisture sensitive reaction, hazardous reaction, transferring the reactant without opening of the reactant vessel. Reaction workup means isolating the product, recrystallization, distillation, chromatographic purifications, spectral data, sample preparations, recording, interpretation. All these are important. Okay. Now you must know the software that I'm going to show here. This is ChemBioDraw. So I can draw the structures, I can modify the structure. So you must know as a chemist, ChemDraw, ChemBioDraw, ACS ChemDraw, anything you want to have, but you should know how to draw the chemical structure. Masternode is the software I can show you here. I'm opening the Masternode software now, and you can see how it is going to be used. Masternode is a software which helps in interpretation. So what I'm opening now, I'm opening from my desktop the files for NMR. Those are called as FID files. Okay, field induction decay files, FID files. And these FID files, when I open, I will go to this FID part, and I can see clearly that these are the peaks which I get, and I can integrate it. I can find out area under the curve for this peak. I can understand whether they are pure or not. Without this software, you cannot handle FID files. Then what you have to do, that you have to just rely upon the operator wherever you submitted the sample. As per his wish, he can give integration. So I can simply give integration on this just by pulling this, and I can find out how many protons are under these peaks. And I can find out whether my structure is matching or not. I can just move down the line and I can find out how many protons. Just I drag and can find out how many protons are there. So this is another tool organic chemistry should have, so that we can interpret our own compounds. We should not be dependent on someone else. And whenever you give sample for recording, ask for FID files and then handle by Masternode. Okay. Now there is another book, very, very important fundamental book, which everyone must have handled, but must have not handled the chapter number 2. Such a long chapter from page number 26 to page number 253. I advise, and I'm sure that this book will be available in the library of every science institute, including pharmacy. And you must go if you want to buy it. There is a link. You can go through. It is costing, I don't know the costing is there or not. It's volume 6, so may not be available. It is 2,000 rupees is the cost for this. This is a different book. It is not Vogel. But you can look at Flipkart and all other things. Find out the source. Very important. I can show you I can just find you for you. Page number 26 tells about general thing, tells about how to calculate, how to keep, how you should be in the laboratory. Okay, running any other hurrying activity is not allowed. It was observed in the undergraduate labs that students are playing with ice, putting that, doing any kind of nasty activity except the practical, and that may be dangerous. Cleanliness of the laboratory, it starts with that. It also tells you what can be the accident procedure, what can be working hours, how you should store your chemicals and the bottles, how the glasswares, waste disposal, how you can plan your experiment, how you can calculate, how you can record your results, the calculations of yield, calculation of everything is given here. Different types of polarimetric data, hazards in the laboratory, explosions and hazards. Everything is given in such books, and I just want to go a little down. I want to show that this kind of assemblies are also given in this book. How to arrange these assemblies, what the components called as, how the addition can be done, how these assemblies, what is the purpose of these assemblies. Everything is given in Vogel book. I expect you to go. I have already shared with Patil sir the book, e-copy of this book, and it will be shared with you. Okay, Patil sir, you can check your email. I just send you the copy. Okay. Now,
Sure,
important
sure.
is documentation. Documentation is important. Whatever you do in the laboratory, whether it's analytical laboratory, whether it's a pharmaceutics laboratory, whether it is chemistry laboratory, keeping data is important. Data integrity is important because in an era of patent, in era of repeatability, the company employs so many employees for the work. If they don't keep data, if they leave the company, where this data will be found, no one will answer later. So there are systems which is called as find molecule. I expect you to go immediately to this site. I'm sharing this DOI, sharing this link with you. You can join this site free of charge. It is written academia, which is free. So you can go through this. I'm just sharing this one. You can go through this site. Okay, find E molecule and go there and search. And you can go to this site and click on academia. So what it suggests you to join, you can try now. You can join for free with no money. There is a video which is given here, which tells you how to operate this. And I can show you, I have my window open here. I can show you how this can be used. Okay. I'm opening find molecule, going to my students actually, sir, this one is available at just $99 per user. So it is around $99 comes to about, if you say $100, 8,000 rupees per user. And we have purchased 6 users. But what we did, we kept the same same user ID password. And how we record our data, I put my initial GUC, I put chemist initial, I put the title, the notebook number, and I put the number of the experiment. And this is my identity, that this is the chemist who submitted this. I want to show you this on my this one. Now, can you see GUC KG? KG stands for Kanchan Gowli, and this is second concept she is handling, and 136 is the practical number of that. This I am just want to show you how am I finding out this. So there are so many experiments are reported. I can just show you how this can be handled very effectively, and you must join it immediately with the free account. You have to just register. I'm just showing one particular experiment here. Okay. So this is recorded. Once you draw the structure, it automatically searches the name and the CAS number and the molecular weight. And if you put here amount of compound being used, it will immediately take 1:1, 1:1.5, whatever ratio you want. So very easily you can handle this. You can put what kind of things you are using, reagents are using, what solvent is used, what is the amount that is used, and what you use, oil bath, water bath. If you just click on this, you can see heating sources, cooling sources are given, and you can type your procedure here very easily. And what we do, I will show you some different experiment where I can show you how we can upload TLC images into this. So you can add TLC images in this software, and you can use them for future. This one, some of them are not reported, some of them are reported. So this is what the software that can be used for finding out this. Now this is the TLC image. I will just make it a little larger. This is the starting material, this is the co, and this is the product. So this reaction I am doing using paranitrobenzaldehyde, malonitrile, using some reagent. I got this product. I used, these are the quantities I used, these are the solvent that I used, and this is the procedure I followed. I can even add Excel sheet into this, so it can be also used for analytical purpose, and I can upload the images of TLC. And I know that 5 minutes, 10 minutes is the time here, and I know that reaction is complete. Starting material is got over. Other starting material is not visible under UV. I can even add NMR files. I can attach a reference file. I can attach everything in this, and this is, there is no limit of I would say online storage available here. There's no limit on this. So for different chemist, I am using different thing here. So I can just go to the other chemist and find out he has reported by different way. Okay. So you can just go to the other chemist and find out how they have reported. Now this is Pawan who has reported this way. I can just go to his site, and he has done few reactions and he is reporting like this. So one chemist can see other chemist data as well, but cannot modify. Okay. So I can go to different chemists and find out what they are doing here, how they are handling it here, and how they are reporting it. So you can find out various things which are done. So this is electronic lab notebook, or ELN, which is called as, and you can report your daily activity. You can be used in future, and most important, for patent filing, they can use this data as a proof that when did you perform this experiment. You can prove it by electronic, because this data cannot be deleted at all. Okay. Now basic technique that you should be mastering is reaction calculations, monitoring reactions by TLC, doing proper literature search, reading the references, and drawing some conclusion, arranging apparatus reaction for various types, isolation of the products, spectroscopic recording and interpretation, reporting or manuscript writing. That is important part, and that's all from my side. So I expect the interactive audience to ask few questions, okay, so that I can answer them. Because I am going to share this, you can get this all links and start using this software so that we can do this. Any doubt, anything you want to more clarification, please ask. This is my email ID and mobile number. You can reach to me by email first, and then if required, we can discuss on phone. For anything, including admissions to ICT or any kind of query that you have in your mind about organic chemistry, I am happy to solve those. So participant, please ask the questions if you have anything.
Dear participants, please, you can raise your questions.
Or if you have poor connectivity, you can put it your questions in the chat box.
Yeah, that may be also possible. Any questions? Dear participants, please come forward. I think no questions are there, sir. You have elaborated it very well.
But anything that comes to their mind, my email ID is anyway there.
Yeah,
Anything
sure,
they want to have link, any problem with the software, especially towards chemistry, not medichem or design. But synthetic medicinal chemistry, I can help them in understanding software. Anything like ELN, I can even arrange some kind of online interaction like AnyDesk or something. And I can explain, I can even give them access with my student, our license version, and they can use that software online. And if find important and useful, even institute can buy that software. If you take more number of users, it will be cost effective, and you can use same kind of password, and you can more users can use that part. Okay.
sure,
So
sir.
any kind of queries, they can send me WhatsApp, they can send me email so that I can answer. Okay. Thank you.
I think no questions are there, sir.
Okay. Okay. So you can close the session.
Sir, actually,
There
one
is some
question is there
there are some
in the
queries
chat box.
coming.
Sir, can we bridge
Can
the
we
gap
bridge gap between drug discovery and its overall development through new advances?
Sir, I was just reading the question. Sir, can we bridge the gap between the drug discovery and its overall development through the new advances?
Yeah, new advances in terms of I'm not understanding if we can clarify. New advances in chemistry you want to say, or new advances in a computational way? I am not clear. The
Yes,
participant.
Patil Gaurav, you can unmute yourself.
Yeah, better.
Yeah, that will
You
be better.
ask verbally will be more clear.
Yes, Gaurav. I have given you the rights to unmute yourself.
Okay.
Sir, he had again just put it some
yeah.
in the chat.
Through the advances in computational chemistry.
Computational chemistry.
Yes, computational chemistry can be used, but I always say that something you have is a tool in your hand. For example, I can write very well. I can with pen which is 5 rupees. I can write with the 1,000 rupees pen. I can write the same thing with 1 lakh rupees pen. Okay, my tool is not important. My skills to write is more important than my tool. Okay, so important part is that whether am I able to understand the science. Tools are just a way to do this. Advances are too many, but the problem is that because of too much automation, we started losing our creativity. So before invention of any computational, even computers for that matter, a supercomputer, which was first made in long back in 80s or 70s. Okay, before that, still the even Second World War time, there were not many electronic devices present. Even a device I'm talking about, forget about computers. Still, there were many discoveries in terms of antibiotics, in terms of other chemicals. Chemical invention was very rampant in Second World War time. Then there were no tools available. It is important to your intuition, important whether you understood biology properly and whether you are able to correlate what you change and what happens. Okay, there has to be some input from chemist side. You should not be dependent on the machine to decide. And unfortunate or fortunate thing is AI. Everybody is after AI, but I'm not, bluntly. And again, already we are handicapped by brain. We are, I mean to say, entire technical education population is handicapped by brain because we are more dependent on design. Can you imagine when there were no softwares? Taj Mahal was built without using CAD-CAM. Okay, the Qutub Minar was built without using CAD-CAM. But the new people with CAD-CAM unable to build, rather the building falls down. Okay, the bridges fall down. Okay, so is it important to have intuition and your own ability? Tools are okay. Earlier there were very less tools. Now there are good tools, but the creativity is going down. So I'm expecting you to understand the fundamentals of thing and then start building your own ability to design. You can take some help. I'm saying it is only 5 to 10% help from the softwares. Rest is your creativity. That is the reason why industries are unable to produce more molecules by systematic medicinal chemistry efforts in last 10 decades compared to earlier era. Earlier era, the discoveries are more in medicinal chemistry compared to post computational era. So there are good examples. Those who advocate it gives very good example, but overall, the inventions were more. And if you carefully look at that, most of the invention, including prontosil, including aspirin, including prontosil to prontosil diet to sulfonamide, including all kinds of natural remedies to chloramphenicol itself was naturally synthesized. Now it is synthetic. Okay. Many discovery came from nature, organisms, plants, and the rate was very high compared to this. But nowadays, the rate is rampantly decreasing for new molecules. Okay. Any supplementary questions you have, Gaurav? So believe in your ability. Yeah. Okay. Thank you. Anyone else you would like to answer? Ask some questions. So any faculty members in the audience? And on analytical side, ICT has NMR 400 and 500 megahertz. If you face difficulty in recording NMR anywhere, I welcome you to submit your samples to ICT. I can get in touch with the operator and you can get FID files at your place.
Yeah, very nice session, sir.
So thank you. But there will be some subsidiary questions. I'm sharing PPT with you. If they come across with that, they can directly read to me. Yeah.
Sure, sir. I will circulate it amongst the participants. Is it okay, sir?
Yeah. Yeah, no problem. No problem.
Yes, sure.
It's all public data. Yeah.
Yeah. Okay, we'll proceed with the next function.
Okay.
Zamir, sir.
Yes.
Shall I stop sharing, sir, now?
Yes, sir. Sure. Please, sir.
Okay.
Dear participants, you can switch on your videos. We can just take some snaps and that will be better. All the participants are requested to please switch on your video. Okay, thank you. Samir sir, please proceed.
Sir. Am I audible?
Yes, sir. Yes, sir.
Yes, you are.
Okay. I welcome chief guest of today's valedictory function, Dr. Ganesh Chaturbuj sir, Professor, Department of Pharmaceutical Sciences and Technology, ICT, Mumbai. Chairman of today's function, Honorable Dr. S.B. Bari sir, Principal HR Piper. Coordinator of today's national seminar, Dr. D.A. Patil sir, Assistant Professor, Department of Pharmaceutical Chemistry. Co-coordinator, Dr. P.O. Patil sir, Head, Department of Pharmaceutical Chemistry. All the dignitaries, dear participants, invited guests, my fellow colleagues, ladies and gentlemen. I am Zamir Khan, Assistant Professor, Department of Pharmaceutical Chemistry, HR Piper, and I heartily welcome you all on behalf of Shirpur Education Society's HR Patel Institute of Pharmaceutical Education and Research, Shirpur, to valedictory function of Kavitri Bainabai Chaudhary, North Maharashtra University, Jalgaon, sponsored one-day national seminar on future prospects in drug design and development. Friends, the journey through life is one that nobody goes through alone. Whatever people may accomplish, those accomplishments were achieved only through the combination of their own personal drive, motivation, love, and support given to them from the people around them. Dear friends, Colin Powell once said, "A dream doesn't become reality through magic. It takes sweat, determination, and hard work." A leader is someone who creates an event team members essentially bring life to it. No event conference can take shape without a hardworking leader. It was the dedication and enthusiasm of our conveners and guides. Their determinations are commendable, and it is their hard work which made this one-day national seminar a huge success. Now I invite coordinator of today's function, Dr. D.A. Padil sir, to brief about the program with concluding remarks. Please, sir.
Thank you, Zamir sir. Good afternoon, one and all. Myself, Dr. D.A. Patil, coordinator of today's national seminar, take this opportunity to summarize Kavitri Bainabai Chaudhary, North Maharashtra University, Jalgaon, sponsored one-day national seminar, Future Prospects in Drug Design and Development. Today, we have started this seminar with the first plenary session of Dr. Sanjay D. Sawant, and the topic of session was design of DPP-4 inhibitors and discovery of sitagliptin. Sir has covered many points through his keynote address as well as the plenary session regarding the time and cost of drug development. Along with, he has focused on the personalized medicines. He has also just put it forward the challenges in the drug discovery and elaborated a case study of anti-cancer drug. Along with that, sir has elaborated the use of innovative technologies like artificial intelligence in the drug design and discovery. Throughout his session, sir has elaborated the anti-diabetics and the number of patients through India, those who are suffering from the diabetes. The actual number of patients is more than the forecast number nowadays for the those who are suffering from the diabetes. And sir has elaborated the DPP-4 inhibitors, their evolution, and the different 17 forms of gliptins and the sitagliptin development. Then the second plenary session was hosted by Dr. R.L. Sawant sir to put light on the topic trends in drug design and discovery. Sir has explained major aspects of research with respect to drug design and discovery. Dr. R.L. Sawant sir has covered many points, including the methods of lead search like serendipity, random screening, and many more. Sir talked about the discovery of penicillins, then tetracyclines, sulfonamides, along with sildenafil, and sir has elaborated the rational drug design with the computer-aided drug design as well. After the lunch break, the third plenary session was of Dr. Ganesh Chaturbhuj sir. He discussed the importance of organic chemistry practices in drug design and discovery. Sir highlighted various aspects in synthesis of new chemical entities, including the focus on basic science and importance of organic chemistry and medicinal chemistry in drug design. And in the day-to-day life, the importance of organic chemistry as well as the medicinal chemistry. Sir has explained the need of mastering of organic chemistry knowledge for the drug discovery. He has also explained the concept of prodrugs and how to avoid the toxicities of drug molecules. He had elaborated the drugs from the natural products like artemisinin, artesunate, vincristine, and vinblastine. Dr. Ganesh Chaturbhuj sir have focused the importance of heterocyclic chemistry for the formation or synthesis of building blocks. Along with that, sir has elaborated the various tools and modules like Reaxis and its uses in medicinal chemistry as well as organic chemistry. Sir has also elaborated the retrosynthesis and the approach and its importance. Sir has also included and explained the Cynthia, the reaction flash, and the other applications like reaction flash, that is an application for the name reactions, and many more like find molecules and electronic lab notebook as well. And with this, I just conclude the summary of today's national seminar. Thank you. Over to you, Zamir sir.
Thank you so much, sir. Friends, it makes little difference how many university courses or degree a person may own. If he cannot use words to move an idea from one point to another, his education is incomplete. Please do remember one thing: we can't climb the ladder of success by keeping hands in our pocket. So, ladies and gentlemen, now I would like to take pause, appreciate who we are and what we have achieved through this one-day national seminar on future prospects in drug design and development. Participants' acknowledgement and appreciation will give us the insight and awareness to move forward towards higher goals and accomplishments. I would like to invite the participants who wish to speak about national seminar. So I request participants to provide their valuable feedback one by one. I request participants to raise hand in the meeting. Or I request a coordinator of today's function to kindly allow participants to unmute themselves.
Yes, sir. Already I have just allowed the participants to unmute themselves. Please come forward.
Yes, Akshay Bhuite. You can give your feedback.
Yes, Akshay.
Hello.
Yes, go ahead.
Hello.
Yes, you are
Hello.
audible. Go ahead.
Yes, sir. Sir, my question is that why is drug design important?
Yes, Akshay. Please, you can repeat your question.
Sir, my question is that why is drug design is important?
Actually, we are not getting your voice clearly. You can repeat it.
Why drug design is important?
Is important, sir.
Why important?
Yeah. Sir, he is asking why drug discovery is important.
Is that question to me?
Drug design.
Drug design is important.
Okay. Shall I answer this question?
Hello.
Shall I answer this
Can you
question?
audible?
Yes, sir. Please.
Why?
Okay. Akshay.
Yes, sir.
Discoveries of the drug are happening by various sources, including natural. Okay. Designing of drug is typically done, especially the rational approach, for the purpose of minimizing synthetic efforts. So you are trying to predict the activity prior to synthesizing. So once you get, suppose you have 10,000 molecules or even 1 lakh molecules made on the computer, not synthesized in the laboratory. When you scan them, during scanning, you will come to know some of them are having some toxic effect. Some of them are having very beneficial effect. Some of them are having better bioavailability. So by screening on a computer, so you can synthesize a limited number of compounds, and then you can reduce your time and amount money to be put in that, and you can come to your desired target very quickly through minimum efforts, minimum money. So once you get a set of molecules, as earlier your lecturers have already mentioned, that how you should come to the lead and then optimize lead further for the hit, and then hit can go to the final molecule for preclinical clinical trials. So it is important to have a design efforts first, but that is not the only tool. Chemist has to put his brain, what would happen based on his experience, real the design to be followed or not. Okay. Any other subsidiary question, Akshay, you have?
No, sir. Thank you, sir. Thank you so much.
Thank you. Someone else have raised the hand prior. Okay. Anyone else who would like to ask or give feedback?
Participants are now open to give their feedback.
Gaurav.
Yes, Patil Gaurav. Gaurav has raised his hand. Please go ahead.
Yes, sir. Sir, firstly, I would like to thank to the Ganesh sir for delivering such a wonderful session. Sir, actually, I have the question regarding that. Can we lead some significant efforts to boost the process of drug development through the Pharma 4.0 or artificial intelligence? Because due to the pandemics like COVID, it is an essential to boost up the drug discovery.
100%. So, the people say that scientists are putting more efforts nowadays. Instead of going from scratch, it is better to go for repurposing. So whatever molecules which are available, which are proven safety, one molecule for one indication can be transferred for other indication through the process of repurposing, and that also uses the tools which are there. Okay. And those, whatever you are using, hello, there is some disturbance. Okay. So these computational tools can also be used to understand the interaction of the molecule which are already invented for one indication can be used for second indication. So you can use all these rational tools to come to point that which could be more effective. So there are many papers coming nowadays regarding repurposing one molecule for others. So you can scan as many as 10,000 molecules and you can get some idea which molecule could be active. If you have biological system available, you can test them. Okay, and then you can repurpose the molecule. I think from R.C. Patel College, Dr. C.R. Patel has published in Nature scientific reports about repurposing omeprazole for neuropathic pain. You can go and see that paper. Okay.
Yes, sir.
Repurposing omeprazole for neuropathic pain. It is actually an antacid molecule on H pump inhibitor, but it can be also used for neuropathic pain. Okay, thank you. Anyone else
Thank
has
you,
a
sir.
question?
Anyone else? I think no more questions are there.
Shall we continue, sir?
Yes, sir.
Now I invite Dr. D.A. Patil, sir, coordinator of one-day national seminar, to propose the vote of thanks for this valedictory function. Please, sir.
Yeah, thank you, sir. Before proposing the vote of thanks, I should express my thanks to Dr. Ganesh Chaturbhuj, sir, who has just taken this lecture with a very short duration of time and delivered a very nice session. Yeah, thank you, sir. Good afternoon, one and all. I, Dr. Dilip Patil, Assistant Professor, Department of Pharmaceutical Chemistry and coordinator of today's national seminar, feel privileged to express my vote of thanks of valedictory function on behalf of HR Piper. I would like to extend my deepest thanks to Kaviyatri Bahinabai Chaudhary, North Maharashtra University, Jalgaon, for providing financial assistance for organizing this national seminar, Future Prospects in Drug Design and Development. I would like to extend my deepest thanks to our patron, captain of our flagship, President, Shirpur Education Society, and Vice Chancellor, SVKM NMIMS, Honorable Shri Amrish Bhai Patel, former Education Minister, Government of Maharashtra, and presently Member of Legislative Council, Maharashtra, for his kind support. I would like to express my deepest thanks to our visionary management, Shirpur Education Society, Shirpur, for valuable guidance and motivation. I am heartily thankful to the resource persons, Honorable Dr. S.D. Sawant sir, Dr. R.L. Sawant sir, and Dr. Ganesh U. Chaturbhuj sir for their enlightening sessions. I also extend my thanks to our beloved principal, Honorable Dr. S.P. Bari sir, for his constant motivation and wholehearted support to organize such events for overall development of students and faculties. I am also thankful to Dr. P.O. Patil, Professor, HOD and Academic in Charge, HR Piper, and co-coordinator for the seminar for his valuable guidance. I am also thankful to all my HR Piper family members, including teaching and non-teaching staffs, students, for their sincere efforts and hard work to make this event a grand success. Most importantly, I am thankful to all the participants of the seminar from the different states of India for their enthusiastic participation. Last but not least, I am thankful to those peoples who are directly and indirectly involved in making this event a grand success. Thank you one and all. Now I declare this national seminar is concluded. Thank you very much, sir.
Thank you. So I will
Dr.
take
Patil,
a
sir,
leave.
to kindly give instructions regarding feedback.
Yeah.
feedback Google form, feedback form, and certificate. I request Dr. D.A. Patil sir to kindly give the instructions regarding feedback form.
Sir, I'll take your leave. I'll take your leave. Thank you
Sure,
very
sir.
much for
Sure.
inviting
Please,
me.
sir. Please.
Thank you.
Thank you, sir. Dear participants, already we have circulated the feedback link in the chat box, and you can fill the information required in the feedback form, and after complete submission of the feedback form, you will get the certificate generated and you can download it. Along with that, there is one important notice that this link will be open only for 15 minutes after the end of this meeting. Keep note this, and you can download or complete your feedback on urgent basis. Thank you.
Thank you, sir.
Any more instructions, sir?
No, sir. Thank you very much.
Okay, I'm just going to end this meeting for all now.
Okay, thank you so much.